Hello. My name is Merrill Asp. I work with Professor Jing Yan at the Yale University Q Bio Institute, and I'd love to talk to you about how bacterial biofilms defend against invasion. All right, so to get started, I want to talk about a little bit of background about the biofilm lifestyle, which is almost one of the most common ways to actually find bacteria living in nature, especially for our model biofilm forming species, Vibrio cholera. So, Vibrio cholera exists in the ocean and in brackish waters where the individual cells swim around until they encounter a suitable surface that might be a nutrient surface or anywhere that the cell can attach and colonize. Once that occurs, a cascade of gene expression changes from the swimming lifestyle to the biofilm lifestyle. And that individual cell and all of its daughter cells eventually build up a biofilm that's scaffolded with the all-important extracellular matrix compounds, which are composed of polysaccharides and proteins that the bacteria themselves produce at great cost to themselves. However, out there in the ocean or in whatever environment the bacteria find themselves, there are other planktonic species that might be interested in the nutritive substrate that they've colonized, and the protection of the extracellular matrix against chemical attacks and physical attacks could be taken advantage of by other invading cells that could, say, attach to the biofilm and incorporate into the community. However, if that were to occur, because the invading cells don't have to take upon themselves the metabolic cost of making that biofilm matrix, they would have an advantage in reproducing and could potentially overtake the biofilm. Now, in practice, that's not what occurs. Um, the biofilms have some method of defending themselves against invasion and attachment by other swimming cells. This has been seen in vitro as well in this study by Carrie Nadel, but the success of the biofilm lifestyle also is testament to the fact that somehow biofilms are able to frustrate the otherwise very robust methods that bacteria have to attach to all kinds of surfaces, whether biotic or abiotic. So that kind of raises our central question of how do biofilms keep other bacteria from attaching to them? We've observed that they can and that they do, but we want to understand the mechanisms that biofilms use to defend themselves from other bacteria in their environment, even of the same species and strain. So the methods that we've used for this, I've been able to leverage the well-developed confocal microscopy techniques that were developed by my advisor, Jing Yan, to observe biofilms at single cell resolution doing Z-stack with confocal microscopy. So this is a biofilm you can see that's mature with a few hundred to maybe about 1,000 cells after a reasonable amount of growth in liquid media. So these biofilms are entirely suspended in liquid while they're growing up from that single seed cell. So in order to be able to image simultaneously the fully developed biofilm and any invading cells that are interacting with it, we developed this hybrid instrument, which we call Holocon, because it combines the confocal platform that images the biofilm with digital inline holography, which is a technique that can image swimming invading cells in an entire three-dimensional volume and reconstruct the trajectories of the cells as they're navigating this complex three-dimensional space that's defined by the biofilm. The principle is actually quite simple. All we have to do is mount in line, so parallel to the optical path, a new laser that's not used for fluorescence, but that's used as a light source. And when that coherent monochromatic laser light interacts with bacteria that are swimming around in our sample, they'll produce a characteristic diffraction ring. And all of the bacteria in the entire volume will produce a diffraction ring. The size of the diffraction ring depends on how far the bacterium is out of the focal plane. So in this holography video, you can see here a number of these different diffraction ring patterns that are moving around that represent different cells that are swimming. And you can see as the size of the diffraction rings changes, that indicates a changing z value. So with each image, we can capture the x, y, and z coordinates of every swimming cell in the entire sample volume. So we can obtain the raw DIH images that look something like this, where you can see the biofilm is this much denser signal, and all of these individual swimming cells interacting with it are also visible. So one important step is just quick background subtraction. We can average together all of the frames in the entire video and subtract those values from the overall pixels, which removes any kind of static signal, in particular the complex biofilm, which would be difficult to reconstruct, which allows us to really focus on the moving cells to see exactly where they are. Now, our collaborator Jiarong Hong, his lab developed a reconstruction algorithm called River, which takes those images where you can still see the diffraction rings and uses a physics-based method that's GPU accelerated to reconstruct what the X, Y, and Z coordinates of the sample that produced those diffraction rings would be. So we get images out of that process that look something like this, where we can much more clearly see the position and even orientation of the individual cells, not only in the XY plane, but also in the XZ plane. And we don't just get these images out, but we get the full set of XYZ coordinates for the entire set of cells, which we can then register with the geometry of the biofilm in order to reconstruct these full trajectories in 3D and really see how the swimming cells are interacting with the biofilm, how their attempts to attach are getting frustrated, which is exactly what we want to see. So, stepping back and looking at individual cells, we've been able to observe a lot of very interesting interactions between the swimming cells and the established biofilms. So what you're seeing here in gray is the fully developed biofilm, and on the left in red, I'm showing the incoming position over a few time points of that initial swimming cell, so that you can see the direction and speed of the cell. But the interaction that we're seeing here with the biofilm of the cell coming in kind of from a horizontal direction along the glass, when it encounters the biofilm, it's smoothly redirected up and away from the biofilm, not really having a chance to adhere. Another significant type of interaction we've observed is temporary adhesion, where in this case you can see a swimming cell coming close to the biofilm, and right here being arrested for a short period of time, but eventually changing direction and swimming away from the biofilm. So this is another type of interaction we see quite often. And one thing that we can quantify is the distribution of dwell times as this temporary adhesion is occurring. How long are the cells staying there? And how common are these different dwell times? So this is something we're still in the process of quantifying. But to understand a little bit more of what's actually happening with the cells and to find ways to change how well the biofilm can defend against invasion, let's look at the motility methods of these cells, not just Vibrio cholera, but other members of the Vibrio genus swim using a single polar flagellum. And because there's just a single flagellum, it's possible for the flagellar motor to reverse direction and for the cell to swim not only cell body forward, but also flagellum forward, just by changing the direction of the flagellar motor. Occasionally, you can get kind of a random redirection in swimming direction, which uh, in these types of swimmers is called flick. So this is the general motility method that you have. Every time the cell reverses the direction of its flagellar motor, it can either immediately reverse its direction or occasionally it will entirely redirect into a new direction. So when we play with this motility, let's first look at kind of the baseline that we have to work with. So this is another assay that we can do where, again, we grow an established biofilm, which you can see here in green. We expose it to a new set of invader cells, which you can see here in red. In this case, when we're looking at wild-type Vibrio cholera, so again, both the biofilm and the invaders are the same species. They're both Vibrio cholera. But when the pre-established biofilm can coexist with these invading cells for about 24 hours, all cells are in a nutritive media, we get a number of attempts at invasion. Now, significantly, this image was also taken after a washing step. So all of the media in the well was removed, fresh media was swept in so that only strongly adhered cells could stay. So we do see a few cells that are attached to the biofilm, but not very many. And the ones that have attached don't seem to have proliferated over those 24 hours. However, if we take a look at a non-reversing mutant, which we developed by knocking out the KY3 from the chromosome, these non-reversing mutants that can't change the direction of their flagellar motor, if they're given the same amount of time to co-culture with this pre-established biofilms, we do see successful invasions. For instance, this cluster here and these clusters here came from individual cells that were able to attach to the biofilm and proliferate enough to essentially take advantage of the biofilm and produce these sub-biofilms within. So this is the opposite of what we tend to see in nature. Usually in nature, the biofilms themselves are able to repel invaders, but with these specific non-reversing invaders, we are able to circumvent the biofilm's ability to defend. So this is very exciting for us. We'll be doing some more work to kind of jump into the mechanism and what's happening with this. One thing that we're trying to compile now that we have the detailed trajectories, we can correlate that with the outcomes after the invading cells and the biofilms can exist together for about 24 hours. And we want to see what types of interactions potentially lead to successful invasion, which type of encounters get frustrated, so that we can understand what the biofilm is able to do, either biochemically or from its topography to protect itself. So with that, I'd like to acknowledge my collaborator, Jarung Hong, the other wonderful members of Yihan Lab, and our funding source from NIH. Thanks so much for your attention. Feel free to send me a question, and I'd be happy to talk to you more about this work. Thanks so much.